So now we can talk about part 2. This is part 2 of photosynthesis. Now the good news is, in some ways, I won't be asking you to know quite as much about it. It's actually still fairly complicated, but again, we're going to simplify it a little bit. So this is the Calvin cycle. Now, in a lot of books, it's called the light-independent reactions, which is really a terrible name. Sometimes it's called the dark reactions, which is even worse. But all of this happens, it's very tightly regulated, happens during light. Because basically, without the ATP and NADPH coming from the light reactions, which are directly tied to light, this doesn't happen. So, I mean, it's just a terrible name. So we just talked about the Calvin cycle. So this, again, is happening in that enzyme-filled stroma, essentially the goo, what would have been the cytoplasm of the cyanobacteria that's now a chloroplast. So it's going to fix carbon dioxide into carbohydrate. P-gal, also known as G3P, same thing. So initial fixation is carried out by Rubisco, ribulose 1,5-bisphosphate carboxylase oxygenase. So it's a carboxylase in an oxygenase. What does that mean? That will actually become important later on. But it starts off with RuBP, or ribulose 1,5-bisphosphate. So that's the substrate for that enzyme. You don't have to really remember any of the other enzymes in this pathway other than Rubisco. So remember Rubisco. Or you could remember ribulose 1,5-bisphosphate carboxylase oxygenase. Okay, so step 1 of the Calvin cycle. And this is probably the most important step for you guys to understand. So we start off with RuBP, also known as ribulose 1,5-bisphosphate. It's called that because, if you notice these yellow things, here's a phosphate, here's a phosphate. Bis means 2, so there's 2 phosphates. 2 phosphates on this particular compound. So it's a 1, 2, 3, 4, 5 carbon sugar. Then we're going to add, notice here, carbon dioxide. So we're going to take some carbon dioxide and water. So this will make a 6 carbon compound, which pretty much immediately breaks apart into 1, 2, 3 carbon compounds. So we've got 2 3 carbon compounds. And this is actually what gives the name to most plants. Most normal types of photosynthesis, we call them C3 because of 1, 2, 3 carbons. This is the first fixed carbon that we have. So you get 2 molecules of 3-phosphoglycerate, or PGA. So we start off with this. This is our 3 carbon sugar. So PGA, then it's going to be added to, you're going to add another phosphate. Notice, where's it coming from? It's coming from ATP. Where did our ATP come from? It came from the light reaction. So this is basically light energy that's been harvested, and now it's being used to carry out these chemical reactions. So it's then converted into P-gal, G3P. This is the end product of photosynthesis. And then eventually those will be converted into glucose, and those glucose units can be linked together to make starch. So a little bit more in depth. So as I just said, we get 6, it's not actually done with 1, it's done with 6 molecules of CO2, 6 molecules of RuBP, again with the aid of Rubisco, ribulose 1,5-bisphosphate carboxylase oxygenase. Resulting complex is split into 12 3-PGAs. We use NADPH and ATP to supply the energy. So the energy actually turns that 3-PGA to 12 G3P. So only 10 of the 12, 2 of them are actually go out of the process. 10 of the 12, we have to make a cycle, and we have to remake that RuBP. Otherwise, this pathway would not be a cycle, and it would just not go anywhere. Because to get from the beginning, we have to get our RuBP back, so ribulose 1,5-bisphosphate. So how does this happen? So here we've got ribulose 1,5-bisphosphate. Carboxylase comes in. We got, carbon dioxide comes in. Rubisco links that carbon dioxide. We get our 3 PGAs. We're going to get ATP coming in. Bisphosphoglycerate. Again, we've got NADPH. And we get G3P, or P-gal. then basically all these arrows here are some fairly complicated restructurings, which are complicated enough that there are enzymes here, but we won't have to learn them. But basically we need to know that to reform ribulose 1,5-bisphosphate, we have to use a little bit more ATP. So to make this back into a cycle, we have to remake ribulose 1,5-bisphosphate.